Its Biomed Cafe menu, Cilia, will be presented today. I will talk about cilia discovery, cilia structure, cilia function, and quiz game and puzzle. I'm Dr. Biomed, cellular and molecular biologist. Hi everybody, welcome to the Biomed Cafe channel. In this channel, complex biological and medical contents are presented in a simple, classified, and summarized form. Cilia discovery. They saw something others didn't see. Cilia was first described in 1677 by Anthony van Leeuwenhoek as a little legs on the protists that he saw in rainwater using a homemade microscope. A sensory function was first suggested for the primary cilium by Zimmermann in 1898. The iconic 9+2 arrangement of the axonal microtubules was first predicted by Manton and Clark in 1952. Satiro and Gibbons in 2014 began to develop the dynein-dependent sliding filament model as the basis for ciliary motility. Cilia characteristics. They extend along the entire surface of the plasma membrane. They are short, slender hair-like structures. They are not found on prokaryotes. They are used to move entire cells, such as paramecium, or move substance along the outer surface of the cell, for example, cilia lining the cells of the respiratory tract. They are complex filamentous cytoplasmic structure. They are used for either motility or as a sensory organelle, depending on their type, and they are often many in number. Cilia types. Cilia can be divided into two groups. Non-motile cilia or primary cilia, and motile or moving cilia or secondary cilia. Non-motile cilia. Non-motile cilia characteristics. They do not beat. They can be found in human sensory organs such as the eye and the nose. They are found on almost all eukaryotic cells, and they are typically singular. Non-motile cilia structure. Non-motile cilia have 4 parts in their structure. First part is axonal region. Axonal region has 3 parts. The first part is ciliary axonum. Its array is 9+0. It means 9 outer pairs of microtubules or doublets. One doublet is A with 13 protofilaments and one microtubule, or B with 10 protofilaments. The second part is ciliary membrane. It's a part of a plasma membrane that surrounds axonum. And the third part is cilioplasm. It fills a space between ciliary axonum and ciliary membrane. The second part of non-motile cilia is transition zone region. It's called ciliary gate 2. It's a porous structure. It controls the flow of molecules between axonum and the cell body. Transition zone region has 2 parts. Transition zone. At the transition zone, Y-shaped structures connect the ciliary membrane to the underlying axonum. And transition fiber. The third part of non-motile cilia is basal body. A cilium is assembled and built from a basal body on the cell surface. It supports cilia. It originates from the centrioles. Its array is 9 outer triplets of microtubules. The fourth part of non-motile cilia is ciliary rootlet. The ciliary rootlet is a cytoskeleton-like structure that originates from the basal body. Its diameter is 80 to 100 nanometers. A prominent component is rootletin, coiled rootlet protein. It's not found in all cilia. Transport of ciliary components. Function of the axoneme in a non-motile cilium. It provides tracks for the motor proteins of kinesin and dynein. Types of transport. Transport of ciliary components can be divided into two groups. Anterograde transport. Kinesin transports ciliary components along the microtubule tracks in an anterograde movement towards the ciliary tip. Retrograde transport. Dynein transports ciliary components along the microtubule tracks in a retrograde movement towards the cell body. Motile cilia. Motile cilia characteristics. They can be found in large numbers. They are mainly present in lungs, middle ear, and respiratory tract. Their length is 1 to 10 micrometers. Their width is less than 1 micrometer. They are also helpful in the movement of the sphere. They beat rhythmically together, whip-like motion, and they form a collective motion that allows fluid transport. Motile cilia structure. Motile cilia have 4 parts in their structure. The first part is axoneme region. Axoneme region has 3 parts. The first part is ciliary axoneme. Its array is 9 plus 2. It means 2 central singlet microtubules, each with the 13 protofilaments, and 9 outer pairs of microtubules or doublets. Each doublet has 1 microtubule A with 13 protofilaments, with 2 dynein arms, is connected to the central microtubules by radial spokes, and 1 microtubule B with 10 protofilaments. They are linked together by nexin. The second part of axoneme region is ciliary membrane. It's a part of the plasma membrane that surrounds axoneme. And cilioplasm. It fills a space between ciliary axoneme and ciliary membrane. The second part of motile cilia is transition zone region. It's called ciliary gate 2. It's a porous structure. It controls the flow of molecules between axoneme and cell body. Transition zone region has 2 parts. Transition zone. At the transition zone, Y-shaped structures connect the ciliary membrane to underlying axoneme and transition fiber. The third part of motile cilia is basal body. A cilium is assembled and built from a basal body on the cell surface. It supports cilia. It originates from the centrioles. Its array is 9 outer triplets of microtubules. The fourth part of motile cilia is ciliary rootlet. The ciliary rootlet is a cytoskeleton-like structure that originates from the basal body. Its diameter is 18 to 100 nanometers. A prominent component is rootletin, a coiled rootlet protein. It's not found in all cilia. Motile cilia movement. Type of movement is three-dimensional motion with a power and recovery stroke. Movement location is axoneme. Fuel is ATP. Ciliary beat has two strokes, power or forward stroke, and recovery or backward stroke. Power stroke. The cilium moves against the surrounding medium with force. This causes the pushing the water backwards and the organism forward. Recovery stroke. The cilium returns to the original position by a flexible movement. Using ATP, the dining arms push on the adjacent outer doublets. Done. A sliding movement occurs between adjacent outer doublets. By anchoring the base of the cilium to the cell membrane and radial spokes and interdoublet links or nexine, sliding is converted into bending. Transport of ciliary components. Function of the axoneme in a motile cilium. It acts as a scaffold for the inner and outer dining arms that move the cilium. It provides tracks for the motor proteins of kinesin and dynein. Types of transport. Transport of ciliary components can be divided into two groups. Anterograde transport. Kinesin transports ciliary components along the microtubule tracks in an anterograde movement towards the ciliary tip, and retrograde transport. Dynein transports ciliary components along the microtubule tracks in a retrograde movement towards the cell body. Cilia functions. Number 1. Cilia play roles in the cell cycle as well as animal development, such as in the heart. Number 2. They also provide habitats or recruitment areas for symbiotic microbiomes in animals. Number 3. Cilia selectively allow certain proteins to function properly. Number 4. Cilia also play a role in cellular communication and molecular trafficking. And number 5. Cilia have also been discovered to participate in vascular secretion of ectosomes. Number 6. Non-motile cilia play a key role as a sensory organelle in the cell signaling, growth control, and energy metabolism. Number 7. Motile cilia are used for locomotion in isolated cells, such as paramecium. And number 8. Motile cilia use their rhythmic undulation to sweep away substances as including dirt, dust, microorganisms, and mucus to prevent disease. Now I want to compare cilia with flagella. Location. Cilia are found in eukaryotes, but flagella are found in eukaryotes, prokaryotes, and archaea. Number. Cilia are one or many, but flagella are usually single. Source of energy. Source of energy of cilia is ATP hydrolysis, but source of energy of flagella is ATP hydrolysis or proton motive forces. Motility. Cilia are motile or non-motile, but flagella are motile. Length. Cilia are short, but flagella are long. Movement. Cilia have back and forth beating, but flagella have rotational or waveform movement. Speed of movement. Cilia are fast, but flagella are slow. And beating. Cilia beat in a coordinated rhythm, but flagella beat independently. Similarities between motile cilia and eukaryotic flagella. They are located on the surface of living cells. Both arise from the basal body. They both outgrow from the cell's plasma membrane. They both have axonym, 9+2 array. Their primary function is for locomotion. And both are microscopic hair-like structure. Thank you for watching. Please subscribe to this channel, and if you like the content of this video, please like it. Now it's time to the quiz game and puzzle. I've put the link to the quiz game in the description of this video. Download it for free and enjoy it. Until the next video, bye!